Welcome back to the channel, everyone. Today we are going to be talking about blotting techniques, and in this video we'll break down the concept of blotting, discuss the three major types of blotting, and explore step by step the process of Western blotting. Now, to begin, what is blotting? Blotting techniques are fundamental tools in molecular biology for detecting specific DNA, RNA, or proteins in a sample. We combine the power of gel electrophoresis alongside molecular probes. Now, to understand the beauty and how blotting works, it is important that you learn about gel electrophoresis, and there is a video on that also on the channel. I'm going to leave a link to it in the description. So if you haven't watched that video, I would advise you check it out first before coming back to this video. But moving on, we're talking about the general principle of blotting. Now, the core idea of blotting is quite simple. Firstly, the biomolecules are separated using gel electrophoresis. So, agarose gel is used in the separation of DNA and RNA, and SDS polyacrylamide gel electrophoresis is used in the separation of proteins. Again, I will advise you watch that video before you come back to this. In the next step, these molecules on the gel are transferred to a support membrane such as nitrocellulose or PVDF. Now, the membrane that has these molecules transferred on them are then treated with probes, which are either nucleic acids or antibodies, which would then bind to the specific molecules of interest on the membrane. And finally, this binding is visualized through a number of detection methods. Now move on to the types of blotting. There are three classical blotting techniques. First is the Southern blot, which was developed for DNA detection. The Northern blot applies to RNA, while Western blot targets proteins using antibodies. And among them, Western blotting is the most widely used in research and diagnostics. So we'll be looking at the steps involved in Western blotting. So step one, the proteins are separated by SDS page, that is sodium duodecyl sulfate polyacrylamide gel electrophoresis. So this separation allows proteins with uniform negative charge to migrate across the gel, and smaller proteins will move faster and they will be lower on the gel compared to the heavier proteins. In the next step, the proteins in the gel are transferred to a nitrocellulose membrane using this setup described here. So you have your gel, this is the gel which contains the proteins, then you put your nitrocellulose membrane right underneath it. You sandwich them in between a pair of filter papers on each side, and thereafter you add foam pad and place all of this inside a plastic cassette holder. And thereafter all of this sandwich is placed inside a transfer buffer, and electric current is added, which will allow the proteins on the gel to migrate downwards from negative to positive onto the nitrocellulose membrane. So this is a simple setup. This is the cassette wherein this transfer sandwich is meshed together and placed into this cassette. This cassette is placed into this holder, and thereafter is placed into the buffer tank where electric current is added or is applied to send the proteins from the gel into the nitrocellulose membrane. So in the next step, the membrane is blocked with a non-reactive protein such as skimmed milk or bovine serum albumin to prevent non-specific binding. What this basically means is that on this nitrocellulose membrane, you have your proteins now bound on it, but there are certain areas of the membrane that the proteins are not bound. So you want to block those areas, and this is very vital for the next step. In the next step, the membrane is incubated with primary antibodies that specifically recognize the target protein for 24 hours. So this is what happens. The primary antibody is bound to the target protein, as you can see, and it wouldn't bind to any other part of the membrane because the blocking agent, the skimmed milk or BSA, has blocked everywhere else. So after that incubation, the excess is washed away so that these free floating primary antibodies can go off. So after washing away excess primary antibodies, the membrane is treated with a secondary antibody which is conjugated to an enzyme. So this is the secondary antibody, and you can see it is conjugated to horseradish peroxidase, an enzyme that has chemiluminescence properties. So this secondary antibody then binds to the primary antibody, and after which the excess is washed away. So it is because of the presence of this enzyme that will allow us to detect this whole complex between the secondary antibody, primary antibody, and the target protein. And this detection is vital for the next step. So in detection method, to detect, we need to develop the blot. And in order to develop the blot, there are different strategies. There are colorimetric strategies where the secondary antibody is bound to an enzyme like horseradish peroxidase, which upon addition of substrate will be converted into a colored product. There is also chemiluminescence detection, where there is a chemiluminescence converting protein, which converts the substrate into a chemiluminescent product. There is also fluorescence detection, where the secondary antibody is fluorophore tagged, and based on the fluorophore, one can detect fluorescence from that sample. Now, each of these methods can be used, and they all have their difficulties and sensitivities. But the most widely used methods are the chemiluminescence detection and fluorescence detection. Now, after detection, the blot is imaged using film or digital systems, and the band intensity corresponds to protein abundance. So, the more intense a band is, the higher the amount of that protein is on the membrane. So these are the applications of the different blotting techniques. Southern blotting can be used for detecting genetic mutations and verifying DNA cloning. Northern blotting for studying gene expression through mRNA levels. Western blotting for confirming protein presence in research and diagnostics, such as HIV or prion testing. Now, in summary, Western blotting remains the gold standard for protein detection, combining electrophoresis and antibody-based recognition system alongside advanced imaging techniques. Now, this tool has been proven to be very vital in modern biochemistry and molecular biology. So this is just a summary of the workflow which you can use to study and remember how it goes. So thank you for watching this mini lecture. I hope you learned a thing or two about Western blotting. Please remember to give this video a thumbs up, subscribe if you haven't already, and let me know in the comment section if you have any questions or you'd like to see a video explaining southern and northern blotting techniques in more detail. I would see you next time. Bye.